Hello, now we are going to talk about the cell cycle, mitosis and the meiosis and the differences and some important points, very small video. First about the cell cycle, we know that G0 is a resting state, then the G1 is their cell growth, and in the S space there's DNA replication. G2 they prepare for the mitosis, so the organelle will be doubling, that will be there. Then the mitosis phase. And one of my lecturer told the easy way to remember is a PMET. What is meant by that? That is PMET mean prophase, metaphase, anaphase, telophase. So mitosis we can remember as a PMET. So G1 is, as I said, it's cell growth. S is DNA replication. G2 is preparation for mitosis. Then the mitosis is the PMET, prophase, metaphase, anaphase, telophase. Then cytokinesis, the cells get separated. Okay. Then cell division is occurring during this mitosis phase, and interphases this G1S and the G2 all together. Now we are going to talk about the mitosis first. Mitosis usually occur in most of the cells. Meiosis mainly occur in germ cells and some other cells. First about mitosis. The thing I want you to remember in this PMET. So mitosis PMET. Thing to remember is M is standing for the coming to the middle, so it's in the middle. A is standing for going away, like anaphase, they are separating. T is for making two. That's how easy way to remember. So in mitosis, in this metaphase, there is no homologous chromosome pairing. Here in mitosis we have 46 chromosomes, and the daughter cell also has 46 chromosomes. There are only the sister chromatic separations occur. So the prophase is there. In the metaphase, all the chromosomes come to the middle. There is no homologous pairing, and those chromosomes in this stage they have sister chromatid. In the anaphase, now the sister chromatid will get separated. In the telophase, they will form two cells, but both two cells have 46 chromosomes, so 2n. Okay. If we go to meiosis, there's something to remember. In this PMET, there are two PMETs. That means meiosis 1 and meiosis 2 is there. And in the meiosis 1, during this first PMET, prophase, metaphase 1, this metaphase 1, there's homologous chromosome pairing. So then PMET, MAT, A is anaphase moving away. In this position, the moving away is not the sister chromatid. The homologous chromosome will get separated. So at the end of the first stage of the meiosis, what will happen is two daughter cells will form, but which contain 23 chromosomes, because homologous chromosome pairs have separated now. Now gene shuffling mainly occur in the meiosis metaphase 1, metaphase and the prophase 1. In the prophase 1, there is a chiasma formation and crossover of the homologous chromosomes, so they will exchange their genetic material. And also in the prophase 1 and the metaphase 1, there is independent assortment. That means the maternal chromosomes or the paternal chromosomes, there is no definite way of division. That means when they are in the metaphase 1, when the homologous chromosomes are standing in both sides in the middle, so it's not one side is maternal, one side is paternal. It's not like that. There's independent assortment. So when in the metaphase 1, after the anaphase, when the cell is dividing into two, there is, for each chromosome there is a possibility of going to one daughter cell. So we have 46 chromosomes, so 2 by 46 times, not 46 chromosomes but 2 as pairs, so 2 by 23 times. There are some more and more possibilities is there to have different type of chromosome combination within these two cells. That is due to independent assortment. 2 into 23 times there are possibilities. Okay, then so mainly gene shuffling occur in this phase. The two concept there is chiasma formation and crossover of homologous chromosomes. Second concept is independent assortment. Now after that it move into the meiosis 2. Meiosis 2 is very similar to the mitosis. So here this diagram I got from Google search. So here in the metaphase 2, it's very similar to mitosis. There is no homologous chromosome pairing further. Here the chromosomes are there with sister chromatid. In the anaphase too, sister chromatid gets separated. Now we have 23 chromosomes inside the cell. Now in the separation we get 4 cells with 23 chromosomes. So all over in the meiosis we get 4 daughter cells. But this is happening in male gametes formation, but in the female female oogonium will produce only one primary oocyte, and then it will produce oocyte, but and other 3 cells here we are telling that we are getting 4 daughter cells, but in female it's only one oocyte and 3 polar bodies. That is just nuclear material only. That's the difference between the male and female in the gametogenesis. Okay. And the cell cycle is under a lot of controls, and when those controls and the tumor suppressor genes, like those are not under control, there's a possibility for going towards cancers formation. We will try to cover it in another video. Okay, thank you.